Thank you very much, Oliver, for agreeing to give a build a cell seminar, and you can get started.
Okay, so Kate, thank you very much for the kind introduction and also for the invitation to give here a talk. And today what I would like to present is our approach to form protocells, really from bottom up. And I mean really from bottom up. And for this, let me start. I would like to show you a little bit the timeline of the formation of our Earth. So it started 4.5 billion years ago, and essentially everything came out of a planetary disk. So this is very well known. And the starting point, chemically seen, is from these gases and a lot of iron. So we had, of course, nitrogen, CO2, CO2-rich atmosphere. Hydrogen was produced by the reaction of iron with water. And we had also gases like SO2, maybe partially in liquid form, and H2S. And you will see later, this becomes very, very important. Okay, then we had the moon forming impact with Theia, and a lot of energy was introduced into the planetary system, which was maybe important because this energy was used to produce also organic molecules. Okay, this is where we start essentially, and we think that everything started with catalysis with iron zero, because a lot of iron was present at this time. And you will also see later that iron plays an important role, for example, with iron meteorites, which entered the Earth. Okay, so we are forming these small organic molecules. Iron became more and more oxidized. More complexity was formed. And of course, this is what you read from textbooks, is that at a certain point, the biomolecular machinery was showing up, explaining essentially the formation of life. Yeah, so with the last or union common ancestor, so forming these protocells. And out of this, bacteria, archaea, and eukaryota were formed. But the big question is really what happened here in between. So starting from these small organic molecules to the biomolecular machinery. And this is where we are targeting essentially our research, and we are looking into these processes that form, let me say, more complexity. Okay, I would like to show you also a historical perspective, somehow in origins of life, because I think it's one of the biggest questions, and already approximately 1,000 or 2,300 years ago, Aristotle was thinking about it, and he coined this term Genesis automatos. So that somehow everything evolved, yeah. They were not thinking, of course, about molecules. They had no idea about molecules, but the idea that somehow out of dead matter, living matter was formed. Then it took really a long time, and there's a German chemist, Friedrich Wöhler. He was the first making urea out of ammonium cyanide. And this is an interesting reaction, and it gives you always the feeling that people were somehow very close to biochemical processes. So for example, here, the ability to make a urea. Then Strecker synthesis, very important to make amino acids. You will see it also later why it is important. Louis Pasteur, he looked into optical activity and discovered chirality on a molecular level. Really a milestone, I have to say. And in 1861, Alexander Butlerov, so this was at the time when people still were tasting the material they produced in the lab, he found that when formaldehyde reacts under basic conditions, sugars are formed. Then a Russian scientist, Alexander Oparin, he coined also maybe this term of a primordial soup, essentially. So he was thinking about an atmosphere rich of methane, which turned out later that it was wrong, and H2S and ammonia and also water. And then I think the important contributions by Watson and Crick and Franklin, the discovery of DNA. And I think somehow with DNA and RNA, most chemists in the field think that somehow, evolution maybe started in the context of prebiotic chemistry. And then the famous experiment of Stanley Miller and Urey, they were simulating a primordial atmosphere, and they found the formation, for example, of amino acids. But I also have to point out that they found not only the 20 canonical amino acids, but more than 200 amino acids, because essentially randomly these amino acids are formed. Of course, the small ones like glycine are formed in larger quantities. Okay, and then Leslie Orgel, he looked into evolution, DNA world, RNA world. Very interesting. Manfred Eigen, he did theoretical studies about also cell formation. You will see it. I think this is very important. And then also he coined this term of a hypercycle. So this was a theoretical model to describe a cycle which has also autocatalytic properties to produce molecules that led to a prebiotic evolution. Very important in this context, the asymmetric autocatalysis, the SOI reaction. So we elucidated very recently this mechanism, which is quite complex. I only want to mention it. Of course, there is no direct prebiotic relevance because this is organometallic chemistry, but it is very interesting because it's the only reaction that works very, very well to show asymmetric autocatalysis with very high enantiomer selectivities observed. And then there was Günther Wächtershäuser. He looked into iron and nickel sulfur clusters. So sulfur chemistry might be very important. And essentially, now let me see, Then in 2009, John Sutherland was showing a new type of RNA synthesis. And I have to say that maybe with this work, somehow this whole field was really encouraged to look again into many, many organic processes that might lead to the formation of relevant molecules that are important for prebiotic chemistry. In 2013, I also would like to mention the work of Jack Szostak, where he looked into RNA synthesis, in particular in protocells, which is very, very interesting. Okay, so I came to this field essentially with meteorites. I bought many, many years ago a meteorite. I looked into the analysis of these meteorites, and I found, and this is very interesting, that these meteorites, iron meteorites, they consist of approximately 93% of iron, 2 to 3% nickel, and 2 to 3% cobalt, and some traces of iridium. And this is more or less a perfect Fischer-Tropsch catalyst. Now, in Fischer-Tropsch catalysis, you start with carbon monoxide and hydrogen, but I pointed out that the atmosphere of the early Earth consisted of CO2. But I came up with this idea: what happens if meteorites, as we also see them in the sky in the night, when they enter a dense atmosphere, they partially evaporate and they are forming nanoparticles. So we started looking into these nanoparticles, and we found that these nanoparticles are active enough to activate also carbon dioxide and convert carbon dioxide into versatile building blocks, mostly formaldehyde and acetaldehyde. So what we are doing experimentally, we started with these meteorites, and this is really the idea that we are looking into these processes from the very bottom up. We are dissolving these meteorites and we are impregnating, for example, minerals, also silica and so on. We reduce it to simulate exactly this process of the formation of these nanoparticles, and then we perform high pressure autoclave experiments and identify the reaction and reaction products that are formed in the course of the catalysis. What is really interesting, this is how these materials look like. So for example, a ball milled Campo del Cielo meteorite is catalytically not that active, but of course, when you are making these nanoparticles, they're highly reactive and active, and they are able to activate CO2. And I think what is interesting, and this was a little bit surprising, so when you are varying here, for example, the partial pressure of CO2 and hydrogen, then you're seeing over a wide range the formation of oxygenated reaction products. So in particular, formaldehyde and acetaldehyde. And I think this is very important because on other planets, you see very often, for example, the formation of alkanes, but alkanes are quite inert. Okay, so there is a question, but maybe I will answer it later, which targets essentially, yeah, let me see, the question here is, do we have a good theory for how this rich, diverse prebiotic chemistry narrowed down to modern biochemistry, pretty narrow in how many amino acids and nucleotides modern life uses? I think I will answer this question later, and you will see that we found and discovered something really interesting that might be an explanation for exactly this question you are asking. So let me continue. I will come back to this a little bit later. Okay, so based on our measurements, so we are doing also kinetic measurements, we can do chemical modeling of a planet. So I call this like an onion-shaped model. Yeah, so we have all these layers in the atmosphere, and we have a reaction network out of these reactions. And because we measure the reaction kinetics, we can also simulate the consumption of CO2 in the atmosphere. And you see it here, and this is very interesting. So what we did here, we varied the amount of meteorites entering our atmosphere. And what is known, and nothing has changed essentially, we have still a flux of roughly 100 tons of meteorites per year. And let us assume 5% of this material consists of iron meteorites, and catalytically active out of these 5% are 1%, 0.5% down to, as you can see here, 0.001%. And this is very interesting when you now simulate this reaction network, then you see that you need a certain amount to completely consume CO2. And this is, I would consider it as a quite rapid process. So within 20 million years, if we have, let me say, more than 0.1% of catalytically active material, we completely convert CO2 into formaldehyde, acetaldehyde, and other alcohols. And what is really interesting, this is, you see here also maybe the first tipping point of our Earth, because the model considers all energy fluxes. And if we do not have enough iron material entering the atmosphere of our planet, then the hydrogen production will decrease, and essentially the planetary system will die in the sense of maybe the formation of organic molecules. Okay, so as you have seen, we know that we are forming a lot of formaldehyde and also acetaldehyde. So we were thinking about what can we do with these molecules, and starting exactly with this starter package. So with the mixture we are obtaining from meteorite catalysis. So initially we started looking into the formation also of sugars, and this sugar network is quite complex. And we looked into all these reactions, as you can see here. It is also possible to do this foremost reaction on anhydrous reaction conditions. So under mechanochemical conditions with a ball mill, for example. So this simulates the process that, for example, water was also frozen, and maybe these aldehydes were somewhere in on the rocky surface of the planet. Okay, so what we see is not only the formation of these sugar type molecules, but also side reactions, so rearrangement reactions, or for example, also Cannizzaro reaction where acids are formed. But it is also interesting, and I would like to point it out, that by these rearrangement reactions, we are getting interesting metabolites, maybe later in prebiotic chemistry, that these molecules like lactic acid or, for example, pyruvate plays an important role in the metabolism of a cell. What is a little bit disappointing, I have to say, is that, and as most people, we also initially looked into formation, for example, of ribose, and unfortunately, the foremost reaction is not very selective, and only traces of ribose are formed. So why is ribose interesting? Of course, because ribose is an important building block for RNA, and the desoxyribose for DNA. So it would be very interesting to have ribose and desoxyribose in larger amounts, but unfortunately this is not very selective. But you will see there is maybe another pathway which has been a little bit overlooked in the past. And this is exactly what you see here. So people proposed the RNA world and the transition from RNA world to DNA world. And there was a lot of work was also done in the past. Here, for example, by Orgel. It's very interesting, like this Orgel pathway forming here these ribonucleosides. The problem here is that under these elevated temperatures and the reaction of ribose, if you have it available, then you are forming the pyruvate form instead of the furanous side form. And this is really a problem. By the way, this was already discovered by Fischer here in Munich in 1917, so quite a long time ago. So he also tried to make these nucleosides, and he figured out that unfortunately always the six-membered ring was formed when he was fusing together ribose with the nucleobases. My colleague here at LMU, Thomas Carrel, he developed another pathway. So where he's starting, for example, with these so-called FAPU derivatives. So this is an open form here of these purine bases. And of course, these are milder conditions because you can form then the aminal with ribose if you have it at hand. And then you can close the ring and form, for example, adenosine. What I mentioned before, and I think this was really a little bit a turning point in the field, then John Sutherland was showing that he was able to make uridine by this pathway, starting here from these amino-oxazole derivatives reacting with D-glyceraldehyde, getting this intermediate, and then the reaction with acetylene nitrile forming essentially uridine. So this was flourishing more or less the whole field because people started looking into, let me say, other maybe neglected pathways to form not only nucleobases, but also amino acids and other molecules which are considered as important in the context of prebiotic chemistry. What people always proposed was that the transition to DNA required the formation of enzymatic reactions. So essentially, this dehydroxylation reaction to obtain the DNA nucleosides. This was very interesting, but I had already in mind that by meteorite catalysis, we are getting a lot of acetaldehyde and formaldehyde. So I had the idea that maybe there is another pathway which is a little bit related to organocatalysis. So where you can, for example, activate acetaldehyde, making the enamine, with a nucleobase, and then let it react with formaldehyde. So this was the idea that I proposed a couple of years ago, and it was very interesting. So when I came with this idea, I looked also in the mechanism, and what I figured out was that if we take nucleobases, and these nucleobases can activate acetaldehyde, forming these vinyl nucleobases, they can stereochemically controlled react with diglyceraldehyde and form immediately under kinetic conditions the furanocide ring and form the nucleosides, the RNA nucleosides. And as you can see here, the reaction conditions are quite mild, so 40 to 80 degrees Celsius. Additives enhance the formation here, and we were able to prove immediately that so these nucleobases can activate formaldehyde, make first the vinyl bases, and then the reaction with formaldehyde or glyceraldehyde results in the formation of DNA nucleosides. So this is quite interesting. So what we concluded out of this is that as soon as acetaldehyde was formed by meteorite catalysis, a pathway was opened for the formation, direct formation of these DNA nucleosides. So we investigated also this mechanism. I don't want to go too much into detail, but it is very interesting that the formation of ribose and desoxyribose is natural. So it is not a selection of the ribose, but the formation of ribose itself is quite selective in such a scenario. Maybe I should mention that people were also thinking about other nucleobases. For example, the so-called Wöhler nucleobases. You have seen Wöhler was the first person or chemist making urea. So he made not only urea, but also biuret, which is like a dimer, the condensation product of urea. And urea can react in a similar fashion with glycolaldehyde and glyceraldehyde, and we were able to show that in situ, also with these Wöhler bases, the ribonucleosides or Wöhler nucleosides are formed. There's a little bit of a discussion in literature going on because these the resulting Wöhler RNA nucleosides are not very stable. They can interconvert and they also decompose. And the question was if they interconvert via a common intermediate. You see here the open form. So we investigated this also kinetically. So we are doing a lot of kinetic studies. We are developing also kinetic tools to investigate quite complex reactions. So here in this case, it is even a simple scenario. You see here, for example, these differential equations, and we can investigate this experimentally. So we determined the reaction rate constants. And what is interesting, and this is surprising, so the interconversion happens not via a common intermediate, but it goes here in a circle. So if you are forming the alpha furanoside, the alpha furanoside is converted into the beta furanoside, and the beta furanoside into the beta pyranoside, and this one in the alpha form, and so on. So it is not a common intermediate, but it goes really in this circle. And of course, the only problem is that this is not very stable, and it unfortunately decomposes. I want to make this short because now I'm getting to the more exciting part. We also looked, for example, in peptide formation, also, for example, in liquid SO2, as I mentioned before. We had a very high pressure of CO2 in the atmosphere, and under these conditions on early Earth, there was liquid SO2 available. So, for example, you can catalyze peptide formation, so amide bond formation with copper. We studied this with all canonical amino acids, and we were able to show that up to octamers, so beyond octamers, the problem is that we are getting more and more an analytical challenge because of all these possibilities of peptide chains, which leads, let me say, to really all kinds of isomers and oligomers. And this is really a problem. But we were able to follow up this up to these octamers, and we identified, for example, the formation of all these peptides. What is also interesting, because there is this discussion if copper was really relevant under early Earth conditions. The reason is that copper typically reacts easily with sulfur, forming copper sulfide. And this is a problem because under these reaction conditions, we need typically copper in a soluble form. But we were able to show that this also works with iron. And the only thing I would like to mention here is that copper shows a very strong Jahn-Teller distortion effect. And probably this Jahn-Teller effect, which is weakly found also in these iron complexes, is maybe the reason for driving these amide bond formations, because the water which is expelled from the reaction is here exactly in this axis. So it forms here a quadratic planar complex, and you have on the tips of this octahedron, you have the water, and water is expelled from the reaction, and amide bonds are formed. Okay, I would like to summarize maybe a little bit this first part here, because you see there are already a number of issues. So, for example, when I was showing you the sugar formation, you see there is no general selectivity in sugar forming reactions. At least the ribose problem is somehow solved. As I've shown you, this is a kind of an organocatalytic mechanism which leads to the selective formation of ribose. We have shown this also in several other cases in the past, but it is a problem. You see also that we are getting only random peptide and nucleotide sequences. So even if we oligomerize the nucleotides, we are getting random sequences. It's not only random, there is also no selective replication of successful sequences. And this is a real issue. Then what I would propose is that we need also cellular structures, that they are necessary because there is a concentration issue. We have to concentrate the organic molecules somehow, because otherwise we are running into a kinetic issue. And I also would like to state that molecules alone do not constitute a self-sustaining system. So we need a real chemical system which is somehow self-sustaining and progressing over time, showing maybe a kind of evolutionary behavior. Okay. Okay, and for this, we made a very interesting discovery. So as I told you, we are starting really from bottom up. And again, we were looking into our chemical starting package. So these aldehydes that were formed by meteorite catalysis. And of course, we looked also into the so-called Strecker synthesis for forming amino acids. Now, if you do this reaction under, let me say, not that clean laboratory conditions, but it's a little bit more dirty, and in the presence of H2S, the surprising result is that you're not only forming amino acids, but you're forming these cyclic compounds. These are imidazolidine 4-thiones. And it took some time until we identified them, that they are formed under these reaction conditions, and they are formed in up to 40% yield. So this is not a side reaction or so, what you are observing here, but these are quite large quantities that are formed in the course of this reaction. And when I identified the structure, I realized immediately that these are really interesting molecules, because this is a prebiotic variant of the so-called Macmillan catalyst. Macmillan and Ben List, they received the Nobel Prize for asymmetric organocatalysis. And essentially what you see here is a real prebiotic Macmillan type catalyst. Okay, so what are the properties of these molecules? It's very interesting. You see here also a hidden amino acid. Yeah, this is the thio amino acid amide, essentially, on this side. What is very interesting, we have 2 chiral centers, so we might are able to control chirality and control maybe homochirality. So this would be an interesting source for homochirality. And we have also shown this, that these catalysts indeed work like the Macmillan catalyst, and they can catalyze enantioselective reactions. So in particular, the alpha alkylation of aldehydes. So this is a classical reaction that was discovered by Dave Macmillan, and he was showing that with his catalyst, so with the oxygen type catalyst, and which is here methylated, that he can control chirality. Furthermore, and this is also interesting, this is not just a normal catalyst which is catalyzing reactions under thermal conditions. It can be also photochemically activated. And this means that this is somehow maybe a first form of a photosynthetic system, because now you are able to utilize the sunlight and catalyze reactions and make products at the end with higher energy. So this is very, very interesting. Here you see the Macmillan type photoredox organocatalysis. So this is the Macmillan type catalyst, and it can also activate, as you have seen, by the way, before with our nucleosides. You can activate aldehydes, you are forming the enamine, but here what makes it special is that Dave Macmillan uses metal complexes, in particular iridium complexes, and with these iridium complexes under UV light irradiation, you can activate other substrates, forming radicals, radical anions, and then radicals, and then you are able to alkylate here, for example, these activated aldehydes. And at the end, you are ending up with modified aldehydes in this reaction. Now let us look at our prebiotic organocatalyst. And here it is a little bit different. So we are starting here with this catalyst that is formed in the reaction, and we can activate in a similar fashion these aldehydes, and they are forming these enamines. And what makes this catalyst now or makes it very special is that we are able to activate this catalyst again also photochemically, and we are forming radicals. So in the first step, we are alkylating this imidazolidine for thione here at the sulfur position, and what we see is a migration of this of the substrate to the nitrogen, and then an alkylation, under photochemical conditions. And what is really special, we do not need a metal. So we don't need a metal because of the sulfur. Sulfur activates this molecule, and we are forming these radicals. And this is very interesting because now we are independent of metals, and we can activate small molecules and modify them. So this is what we have shown in the past. Okay. I also would like to show you that there is also a rare property. They are forming enantiopure crystals. This is a very rare property because if you, for example, try to or crystallize racemic amino acids, you have to seed it, for example, with enantiopure crystals, and very often you're forming just the racemic crystals. Here in this case, you can seed it, and you're forming enantiopure crystals, and this is very interesting as a starting point for development of homochirality in the context of prebiotic chemistry. Okay. Now, I've shown you that under these little bit dirty Strecker type conditions, we can form these organocatalysts, and these organocatalysts are able to modify their own building blocks. Yeah? They are forming a second generation aldehyde. So we are able to modify our own building blocks. And we looked into this reaction cascade. And first of all, what I can tell you is that we observe the formation of 11 out of the 20 canonical amino acids. So under these organocatalyzed reaction conditions, we are selectively forming canonical amino acids. Not as you have seen, for example, before, like in a Strecker synthesis, where you just get random amino acids, or for example, in the Miller-Urey experiment, where you can detect many other amino acids. Here in this case, we have selectivity for the formation of these canonical amino acids. So we investigated also this mechanism by kinetic studies, and I told you already that this is very different to the Macmillan type organocatalyst that we see in the first step an alkylation of the sulfur, and then of course the formation of this enamine. We have also crystal structures of it, so we can really prove that we are forming these intermediates. We can isolate them. We can follow them by NMR studies, doing kinetic studies. And what is interesting, and I only want to summarize it here. So this is again here the Macmillan type organocatalyst. Here is another famous organocatalyst. This is the so-called Jorgensen organocatalyst. And it's interesting, our prebiotic organocatalyst is exactly in sense of the nucleophilicity between these two catalysts. So closing somehow a gap, an interesting gap in catalysis. So to make a long story short, what we have seen is, and this is a little bit surprising, because normally as a chemist, you are making a catalyst and then you are catalyzing a reaction. Here, in these systems, we observe something totally different. We observe dynamics in the system. we see that the first generation catalyst that is formed is catalyzing the modification of its own building blocks. And now there are two pathways to generate a second generation catalyst. And this happens in the reaction mixture. So this is not something we have to force. This happens over time. So we only observe the reaction mixture. And there are two pathways. There is one pathway that we form the de novo, again, a new second generation catalyst, or the other pathway is that this group here on this side here gets exchanged by the newly formed aldehydes. And we are getting another second generation catalyst. So we investigated this in detail. And what we observe is a real molecular evolution. So we are starting with our pool of aldehydes and ketones, which we obtained by meteorite catalysis. And now we are forming, just under these a little bit dirty Strecker-type reaction conditions, our first generation catalyst. And this first generation catalyst modifies its own building blocks. And over time, we see the formation up to a third generation type of catalyst. And this evolutionary behavior depends also on the chemical environment. So depending if we have ammonia or we have no ammonia present, for example, then we are seeing here the formation or the enrichment of certain catalysts. This is quite interesting. Let me see that there is another question about initial conditions. Okay. Yeah. This is a very good point. So the question is here, under different initial conditions or due to stochastic nature of chemical biological evolution, we would have different world if Big Bang started again, right? Or other planets with living systems. Okay, what is your opinion on this point? Yeah, this is maybe partially true, but I think what we see here, we see an intrinsic property of these catalysts that are formed. And of course, there is a certain variation. Yeah, there's a variation which is influenced by the availability of these aldehydes and ketones, which will lead to maybe some or different diversification of these molecules. But essentially, we see an intrinsic property in the formation of these catalysts. And it is a little bit a difficult question maybe to answer, because this would really require to investigate very, very different starting conditions. But at the moment, we see, let me say, a quite good stability in the system, because we are forming a lot of formaldehyde and acetaldehyde at the very beginning from this meteorite catalysis. And this leads to a limited set of these organocatalysts. And I think this might be an explanation that we are not getting a huge diversification of these catalysts, but a quite stable system, which is maybe driving into one or into a certain direction, maybe, depending on the environment, as we have seen, for example, by varying ammonia concentrations and things like this. A little bit difficult to answer, but yeah, maybe we can discuss it later. Okay, so this leads to some Darwinian evolution. And by the way, this catalyst is not only modifying the catalyst itself, but it is also catalyzing important reactions. So we have identified that this catalyst can reduce selectively, do phosphorylations in the 5' position here of these RNA nucleosides. So we are obtaining the phosphorylated nucleotides in the 5' position with really great region selectivity. By the way, I mentioned that this catalyst is also a photocatalyst, and what we see is that we can not only phosphorylate, as you have seen, these nucleosides to get the nucleotides, we can again also activate these nucleotides with a catalyst here in this position. And then this is very interesting. This molecule is really stable over long time. But by irradiation, for example, with UV light and other nucleotides, we see the formation of oligomers. So here it would be a mixed oligomer consisting of a DNA building block and an RNA building block. So this is also quite interesting. What we also did, we used this chemistry to make NAD+. So it's very interesting. Orgel many years ago proposed here a pathway to NAD+, but essentially only the first steps were possible to realize. Now we combined here our selective formation of ribose with exactly the formation of the building blocks which lead to these nicotine amides finally. So as you can see here, we can make the amino ribose here in this case, and then we fuse it together with the chemistry of Orgel, and indeed in situ, these nicotine amides are formed. These nicotine amides, they can be phosphorylated, selectively phosphorylated with this catalyst. And then by this photochemical reaction, we fuse it together with the activated form of adenosine nucleotide, and we are getting at the end NAD+, which is a quite interesting reaction. Okay, now I'm coming to something that initially would be quite obvious, and the obvious question is, if you think about organocatalysis to catalyze aldol reactions, and but I want to show, before I'm going to this, I want to show you some other work from John Sutherland. So John Sutherland looked, for example, also in the formation of lipids, and what he did here was starting essentially from HCN, so making these nitriles. These nitriles can react again with H2S or HS minus, also to these hydroxylated thioamides, and these thioamides under UV irradiation, they form acetaldehyde. Yeah. So we can shorten essentially this synthesis. I've shown you that by meteorite catalysis, we are getting a lot of acetaldehyde. So we do not need to go this pathway starting from HCN to make acetaldehyde. We can make acetaldehyde directly. But what was quite interesting, so he did a classical aldol reaction under basic conditions. And under these basic conditions, typically you get the normal aldol product. Now, he reacted it for 34 days, so quite a long time at room temperature. And then under these conditions, he observed up to the C8 aldehyde, yeah, elimination of water, you are getting these unsaturated aldehydes with up to 12% yield. So I have to say this is not a very good yield, but very interesting. But now again, he was thinking about it to make out of these lipids. And what he did, he hydrogenated these unsaturated aldehydes with Rana and nickel. So this is not maybe a real prebiotic chemistry, but you can do this, and then you are getting the saturated aldehydes. And of course, in the next step, you can react these, you can oxidize them to the corresponding fatty acids, so short chain fatty acids like the octanic acid, for example, or the aldehyde, or just by dispropionation, you can also get the alcohol and the acid. So this was the starting point for this type of chemistry. And then he looked into another building block. So taking phosphorylated glycerol, and of course, you can activate this phosphorylated glycerol and acylate it. So acylate it here with a short chain carboxylic acid derivatives, and then you're getting kind of lipids. And there are two pathways. Of course, you activate then the carboxylic acid derivative, you activate it, and you see or observe migration, and you get the acetylated glycerol product as a lipid forming molecule. So he was looking into this, and indeed this works. And you can imagine, of course, you are forming these lipids, and these lipids, they self-assemble then in kind of cells or micelles, initially micelles, and then maybe protocells. And they looked into this formation of these protocells with this, more or less, I would say artificial system, because you can imagine it is really tough to get here these long chain lipids, and it ends somehow with C8. So an elongation of these lipids is really difficult. Okay, so, and this was a little bit the starting point for me again, because I was thinking about it. We are producing a lot of acetaldehyde, and acetaldehyde can be activated with these organocatalysts, as I've shown you before, and so an aldol reaction should be possible. So we did this, and initially we thought it's really strange. We are not only getting the normal aldol reaction products, but what happens, and this is what you see here in these vials, you are getting always a milky solution. And this milky solution comes from not only the normal aldol reaction, but what we observe here is something totally different. We see an aldol oligomerization reaction. So it doesn't stop also at the C8 oligomer in this case, but it continues. And this is very interesting. So it continues until these lipids that are formed, they form droplets and lipid droplets. And this is what you see here. And you see also that this reaction compared, for example, to the Sutherland system is quite rapid, because we see this formation of these lipids already after a couple of hours. So our organocatalyst is totally different to the use of normal bases. And this is essentially what I would like to show you here, that we see a simultaneous formation of lipids and protocell formation. So here, in this research, we found that acetaldehyde gets activated, and acetaldehyde in an aldol oligomerization reaction forms these lipids, and these lipids, they undergo also different reactions. They can, for example, also exchange now again with our catalyst, like in this evolutionary behavior, that we exchange here the residue R1 by the newly formed aldehyde. And this means we are getting lipids which are modified by our organocatalyst. I want to show you here a video, and this takes only 2 hours. And this is really exciting because initially you see almost nothing. So this is a reaction, you do it under the microscope and you just observe it. And now you see already these a little bit dark yeah, circles here forming, showing up. These are droplets. These are like oily droplets that are formed by the formation of this aldol oligomerization product. And this is, maybe I show you, let me see, maybe I can show you the time scale. No, it's a little bit, I can control this. So after 1 hour, you see already the formation of bigger structures, and these are really looking like cellular structures. Maybe I jump here to the, oops, I want to show you, yeah, here I can control it, yeah. I want to jump here to the end here after just 2 hours, and you see that this is really an exponential growth of these protocells that are formed in the course of the reaction. And what we did, we coupled our microscope essentially to an Orbitrap mass spec, so we can observe and watch the formation of these oligomers in the course of the reaction. And for example, here we varied the acetaldehyde concentration, we varied the catalyst loading. And of course, you can imagine at higher acetaldehyde concentration and catalyst loading, we are getting more rapidly the formation of these type of protocells. But what is really interesting is, this is what you see here on the right side. This is the formation of these lipids. And you see here the total chain length stops at approximately C16, C18. Yeah, this is very interesting. And what we also see is, we see not only the formation of the normal aldol product, so the formation of these alcohols, but also the unsaturated system. So this means that water is eliminated from the reaction product, and we are getting these unsaturated lipid chains. And not only this, we are getting in situ the formation of these lipid chains and the protocells. So they assemble themselves into these protocells, and you see it here. So after 1 day, yeah, we have a distribution here, total chain length of up to C20, quite long chains. We investigated also this reaction by dynamic light scattering, and it is very interesting. We see here the formation really of these larger protocell type structures. Also, a question that always comes up is the salt tolerance. So we see that the formation here of these protocells tolerates really salt additives. So we did the reactions, for example, with 200 millimolar sodium chloride and 400 millimolar sodium chloride. And you see that essentially, of course, the formation goes a little bit down, but it is still quite stable even at these high salt concentrations. I jump here further. I want to show you another video, which is also very interesting to show what happens in these initial droplets that are formed. So what we see, and these are the dark spots, this is water which is expelled by elimination of water from these lipid chains. And we see also sometimes really these water droplets popping out of these catalytically active oily droplets. And at the end, what happens is water is accumulated inside, and they are forming protocells which are filled with water. So as I told you, we are watching, for example, the formation of these protocells by a mass spec. So we can identify all these reaction products. So these are only the first initial steps, and the chain is growing and growing. So, for example, we also isolated intermediates here, showing you that these newly formed aldehydes, they exchange, for example, also with the acetaldehyde moiety of our imidazolidine 4-thione catalyst, and they are forming new catalysts. And this is very interesting because now you can imagine the catalyst gets attached to the surface of these lipids. So this is a summary of the reaction mechanism, what happens here in these cells. So starting from acetaldehyde, we are forming the normal aldol reaction product, but this aldol reaction product reacts like in the fatty acid synthesis, very similar, in a very similar way. It extends it, and we are getting these aldol oligomers in the course of the reaction. And so we are getting these long-chained aldol reaction products, and these long-chain reaction products, they eliminate water, and they are forming then also the unsaturated aldehydes, and these aldehydes, they can exchange with the catalyst and attach the catalyst to the surface of these cells. So this is what we see. So it's quite interesting. So this is, for example, a droplet formation, initial droplet formation at the very, very beginning. And then we see this elimination of water. So the droplets that are coming out. And at the end, this is the final product. Yeah? So this can be formed within 2 hours. We are seeing an empty cell, which is filled with water. And for example, by staining these cells with dyes, we know also that, for example, organic molecules are accumulated inside these protocells, and these organic molecules are then activated, and they can react, for example, to these aldol oligomers over time. So this is very interesting because we think that we are somehow overcoming a little bit the concentration problem, because the high, let me say that the low concentration of organic molecules just in water. Here in this case, we are accumulating organic molecules inside our protocells, and this enhances then at the end also the reaction to other interesting reaction products. One thing I would like to mention, because these are now the next steps, we are looking into it, and it's a little bit an outlook. What we also found is that our organocatalysts, they are modified, they are ribosylated by the same process as I've shown you before, by reaction of glycolaldehyde and D-glyceraldehyde. We are getting here these furanosides, which is quite interesting. And if you think about it, our organocatalyst can also be considered as maybe a letter code. So we can make, for example, these catalysts just from formaldehyde, then they are unsubstituted, or formaldehyde and acetaldehyde, and then we are getting here methylated, a methylated organocatalyst, or this would be another constitutional isomer here. We are forming the methyl group on the other side, or the dimethylated, and of course there are a number of stereoisomers that are also formed. So we are looking into this process, and I think this is quite interesting. You see, this is not just a normal catalyst maybe, but it can also act as an maybe information storing molecule in this sense. Okay, so we are thinking a lot about kinetics, and what we see is that essentially the system is totally kinetically driven. So what I've shown you here is the formation of these aldehydes, and these aldehydes, they form the first catalytic system. So it generates the first catalytic cycle, and you can now imagine that over time, because of this evolutionary behavior, we are entering in a second generation cycle, a third generation cycle, and so on. And this is very interesting. So mathematically, this can be compared to a hyperbolic spiral, which is also, for example, found in galaxies. And so we are thinking a lot about this kinetic behavior in this reaction. So we are interested in these reaction networks, also autocatalytic subunits here, as you can imagine. And we can also separate somehow these subunits by what I consider as communication layers, that they communicate between these cycles and drive these reactions. Okay, with this, because I'm almost over time, I would like to summarize my talk. What I've shown you is we are starting really from very small molecules, just from CO2 and hydrogen. And by meteorite catalysis, we are getting the first building blocks. And what is really exciting, what we were able to show, is that we can form maybe first organocatalysts, and that these organocatalysts, they undergo some evolutionary change or behavior, and they are forming other catalytically relevant systems. And finally, what I've shown you, that they even drive the formation of these lipids, and that these lipids assemble in a self-assembly in these protocell structures, which are organocatalytically modified. And you can imagine that these protocells are catalytically active and can drive all kinds of reactions, like phosphorylation reactions and things like this. So we are looking into this, and this is a quite exciting field we are working in at the moment. So I would like to thank my group, and I also would like to thank all these funding agencies for the great support, financial support, and you for kind attention. Thank you so much.
Thank you. Thank you. That was amazing. That really tickled my prebiotic chemist.
Yeah.
Um,
Sorry,
there is-
maybe it was a little bit too much and in the short time, but it's yeah. Okay.
There's no such thing as too much of prebiotic chemistry.
Okay. Mhm.
There's many questions in chat.
Okay. Maybe I continue here. Okay. Yeah. Okay, there's one question. Does this work support eddy processes, dynamic kinetic stability theory for origins? Yes, I think so. I think so. I think so. I still have to point out that we are still at the very beginning with all these processes. It is a really difficult system, and sometimes we are only the observer of this system. So we trigger the system and then we observe it. We just let it run and observe what comes out. And I think there are many facets that are highly interesting. So there is autocatalytic behavior in the system. There are many kinetic or dynamic kinetically controlled processes involved, and we will try to elucidate them all. But it is really a tremendous work, I have to say. Yeah. Mm-hmm. Okay, now let me see. I hope I answered a little bit your question. So this is at the moment we cannot really predict what happens and how this system changes over time. But we are doing experiments, and we are about to establish, for example, also a small metabolism. in these protocells, because you can imagine we are forming, I pointed it out, we are forming, for example, also lactic acid and pyruvate and things like this, and we have already NAD plus in the system. So you can imagine we are working exactly on this to establish also metabolism in our protocells that are formed. Let me see.
I think there was a question really early. Where was it? You answered a question that was below it from Hans, but there is or from Adam, but there's a question from Joanna at 11:11. Can you see that? I think you moved past that.
Here. Is it this question here? Why is organometallic chemistry irrelevant prebiotically? Or did I misunderstand the reference around the soy reaction? Is this the question you're asking?
Yes,
for?
yeah.
Okay, yeah. The problem is organometallic reactions are sensitive to water. And this is a problem in general for prebiotic chemistry, because we can assume that all these reactions somehow only work in water. And typically, for example, the soy reaction, you need anhydrous toluene, because otherwise this reaction would not work. So even if you use, for example, ethers, so more polar solvents, the problem, for example, in the soy reaction is that you are not forming these agglomerates, like dimers and things like this, which force the reaction to get an imbalance, a chiral imbalance, which is then amplified in the course of the reaction. So I think it's a little bit difficult to explain, but for example, in the soy reaction, people always assumed that only these zinc alkalates are the catalyst, but this is not true. The reality is that these zinc alkalates in the soy reaction, they form with the aldehydes hemiacetals, and these hemiacetals, they are quite stable under anhydrous reaction conditions, and then they amplify the chirality. So this is the mechanism in the soy reaction, and this is why I think that the soy reaction itself, of course, does not play any role in a prebiotic context, because you're forming reaction products that are quite limited in the substrate scope, I have to say, and they are not really related to the molecules we know, for example, amino acids and things like this. So it is maybe a target to develop asymmetric autocatalytic reactions that show a broader substrate scope, and maybe also that can be applied to other reactions. So this is quite complex, yeah. I hope I answered this question. Now, there were many more. There's one question: given the current climate crisis and pollution, what types of chemical reactions will be possible, and what effects those reactions will have on biology and humans in the future? Yeah, difficult to say, but I also would like to point out that maybe the starting reaction conditions on early Earth were very different from the reaction conditions we see nowadays. We have a lot of oxygen, so oxidative reaction conditions. So unfortunately, I have to say that we cannot use the reactions we see, for example, by meteorite catalysis to convert CO2 into versatile organic molecules, because nowadays, of course, these meteorites, they get just oxidized and they would form iron oxide, and iron oxide is catalytically more or less dead. So this is the problem. Yeah. So Yeah, this hard to say. This is also a kind of an evolution where the chemical reaction space maybe has changed over time. So nowadays we have a different reaction space compared to the reaction space at the very beginning on our early Earth.
So Kate, may I jump in?
Mm-hmm.
So wonderful talk.
Yeah.
I really enjoyed it's kind of, refresh my chemistry 30 years ago background. So this is fabulous. So I got basically combining my two questions I asked in the chat. One question is about origin of life or the evolution of chemistry and the biology, and then second question is the future. So the as I mentioned earlier, if we start Big Bang again, I do believe, we have probably different chemistry, like, I don't know, 21 amino acid or different nucleotide. And I also do believe in the aliens because that is a natural kind of, the conclusion I had. I just wanted to have your a little bit more elaborated kind of opinion on, the stochastics kind of reaction system and condition a little bit, different in another planet, etc.
Okay, so what we see, and I think this is in some sense very interesting. This organocatalytic system gives more selectivity. This is very different to what we would see if we would just mix components together without such an organocatalyst. So initially, what we also thought, we would expect maybe an explosion of reaction products, various reaction products. But this is not the case. So we see, of course, as side reaction products, maybe elimination, rearrangements, things like this, giving a broad range of products. But the main path is somehow completely controlled by these organocatalysts, and they give high selectivity. And this is what we think. So this is our approach. What I pointed out at the very beginning, we are starting really from bottom up. So we are starting with the starter package, because everything else, if I take just the chemicals from the shelf and I mix it together, this is not the real system, which might lead to a different outcome. But we are really taking this mixture of organic compounds we obtain by meteorite catalysis, and then what we see is that that not really an explosion of a variety of components arises, but there is a high selectivity in the formation of certain molecules, and this is what we try to understand at the moment.
So in that case, I mean, let me kind of rephrase my question. So because Kate is here, so in a different condition, in different planet or another side of the universe, we may have mirror image world completely, chirality is completely opposite, compared to the Earth. Is it possible, right?
This is possible, and this is essentially, this is also our target. So what we try to do, or what we will do in the future, is we want to have not our catalyst derived from L amino acid, but with a D amino acid, to see if we are forming L sugars in this case, or the opposite chirality, because otherwise I would say nobody will believe that we are making protocells, which, by the way, if they are forming, for example, branched lipids, they are also chiral, and they will have probably exactly the opposite chirality. So this is what I would expect if you are starting with D amino acids, forming L sugars, and so on. So generating a mirror image world in this sense. So this
Okay,
is,
thank you so much.
this is what I would like to study, yeah, to understand what really happens.
Okay, thank you. Wonderful.
Yeah.
Thank you. There's another question on the very bottom of the chat from Joanna. I think that's a follow-up to what you answered.
Okay. Okay. I'm always really okay. Okay, a model system. Yeah, that's right. Okay. The more general irrelevance of organometallic chemistry to water, I'm not thoroughly convinced. Okay, 55 molar of water is the laboratory reality, not that of cytoplasm. Okay, lots of people have been thinking of low water activity, chemistry, mineral surfaces, and so on. Okay, yeah. Difficult to answer. Yeah, maybe if you have special reaction conditions, or for example, even supercritical solvents, things like this. Yes, then in this case, I would really maybe consider also organometallic chemistry. So why not? But in a pure water system, it might be difficult. Except you are using palladium or other transition metals, where you can also achieve maybe transformations, organometallic transformations, even in water. But if you are thinking about organometallic chemistry with Grignard reagents, zinc organoles, and things like this, this will be really tough to realize it in water. It's impossible, essentially. So, yeah. So I hope it answers your question, Joanna.
Yes, thank you so much, Oliver. Thank you.
Yeah. Sorry, it's a little bit difficult to explain. I would love to have a board to draw some chemistry there.
Yeah. Okay. Honestly, I kind of lost track. Is there any other question you didn't answer?
Let me see. I also checking.
Let's see.
There is another comment or question. Let me see. You mentioned Sutherland's work. His pathways often use compounds like cyanamide, glycolaldehyde, cyanacetylene. I agree each molecule can be plausibly produced, but likelihood of all appearing together in sufficient concentration at the same location and time is uncertain. Yeah, I totally agree with you. And some intermediates like cyanoacetylene are highly reactive. Absolutely, I also agree. So if you have worked once with this molecule, you know that you have to store it in a deep freezer, because otherwise it polymerizes. It's terrible. Do you think this is really prebiotically plausible pathway, or we still need to do more work on validating and making prebiotic plausible pathways? Yes, I agree. We have to work on this, and maybe I also would like to give you another perspective, because very often HCN is discussed. Now, HCN at high concentrations is also difficult in the atmosphere, because it will polymerize or decompose. But now again, our meteorite catalysis comes in. Imagine you are forming HCN in the atmosphere by photochemical processes and so on. HCN will immediately react with the aldehydes, and we have shown that they are formed with these aldehydes to form cyano alcohols or aminonitriles. Sorry, aminonitriles. And this means that HCN reacts immediately with these aldehydes, because they are reactive enough to form somehow a stable form of HCN. Yeah, it doesn't mean that HCN is gone. It that it reacts and the reaction is over, but you can form intermediates which are stable enough over a long, long time, but they can still dynamically exchange and release HCN and react with other molecules. So this is not a dead end. I think this is very important also to understand that prebiotic chemistry, and this is also very often overlooked, that prebiotic chemistry is a lot about the dynamic formation of molecules and dynamic decomposition or rearrangement to other molecules. And it is not just this pathway to a single molecule we desire to make. This is very, very important. And I think to better understand this, we really have to investigate these reactions. These are complex reaction networks, which you can only understand by, for example, kinetic measurements. And with this kinetic data, then you are able, for example, to simulate other scenarios to make predictions where molecules like HCN end up at the very end. I think this is very important. But I totally agree that I also have my doubts with these highly reactive molecules when I see, for example, the formation of acetylene and other molecules, which are hard to stabilize. So this is, I totally agree with you. Yeah. Okay.
Thank you. Thank you. I think you answered all the questions now. Thank you. Thank you so much.
Okay. Thank you. Thank you.
And thanks everyone for a great discussion. That was a real treat for the chemist in me.
Thank you so much. Thank you.
Thanks, and have a great rest of your day.
Yeah. Okay. Have a nice day. Yeah. Thank you. Bye.
Thank you. Bye-bye.